Hello everyone and welcome to this MAST webinar. My name is Bethan and I'll be chairing the session today. The title of today's talk is Genetic Mechanisms for Acclimation and Adaptation to Coral Bleaching, and that is with Sebastien Leveque from the University of Glasgow. So just a couple of housekeeping notes to begin with. After Seb's presentation, we'll have a live Q&A session. So if you would like to ask a question, please use the Q&A box that you see at the bottom of your screen, and you can input a question at any point, but we'll work through those questions after the presentation is finished. So this session is scheduled to end around 1:30 p.m. UK time, but we may go over due to questions. And just to let you know that the entire webinar will be recorded. So if you have to drop out and you miss something or you can't stay for the entire session, you can find this recording on the MASTS YouTube along with all our previous webinars. So a quick introduction to today's speaker, which is Seb. So Seb is a year and a half into their PhD with the University of Glasgow, but they're currently based in Sweden where their supervisors are based. His research investigates coral bleaching trajectories in the context of climate change. And before his PhD, Seb did a marine biology degree in France, where his thesis focused on corals in Singapore. Just a reminder that the work that Seb is talking about today was recently funded by the Saltire Emerging Researchers Scheme, which was a joint collaboration between the Scottish Funding Council and MASTS. So that's enough from me. We're going to hand over to today's speaker.
Thank you, Bethan. Hello, everyone. So here's my webinar. Like Bethan just told you, we will talk about the different mechanisms around acclimation and adaptation for coral reefs, especially to coral bleaching. But first of all, I want to take a few minutes to explain to you what are exactly coral reefs. So coral reefs are one of the most diverse ecosystems in the world, where 25% of all marine species live in and around the coral reefs. As you can see on that picture, coral reefs are quite large structure, but they are made of really small animals called polyps. This animals is primarily responsible for building the reefs, as you can see on that picture, and can take many different forms, like large reef buildings, graceful flowing fans, and even small and solitary organisms. And it represents around 1,000 different species that have been discovered from now on. The specificity of coral reefs is that they live with algae in a type of relationship called symbiosis. This means that the organisms cooperate with each other. When the algae called symbionts live inside the coral and got a shelter from the coral, in return for that, the algae provide to the host with food that produced through photosynthesis, and it can represent to 90% of the metabolic rate of the coral reefs. So this specific symbiotic relationship that will happen between the algae and the coral itself is strongly dependent on different factors, such as pollution, light, and one of the most important one is temperature. And when I say temperature, it is refers to the global warming situation, where we see a lot of sea surface temperature rise. And this phenomenon called the symbionts to be expelled from the coral tissue, causing its loss of color for the corals and the major source of food that occurs within. And this is what we call coral bleaching, which turn the coral to like white patches, as you can see on the left picture. But there is also good news: is coral bleaching does not always mean the death of a coral reef itself. Coral reefs can recover their symbiote algae in time, but the process requires cooler temperature and less impact of pollution or ocean acidification. And from that specific phenomenon of coral bleaching, our project aimed to explore the baseline variability and density of this symbionts community within one species of corals during the bleaching season. And from that major goal, we found 3 different questions we wanted to explore. First, are there distinct spatial patterns in the symbiont density and community composition in a non-bleaching year? And first, are these patterns also got a temporal aspect within the symbiont density and community composition as well? And the third one is to explore if the symbiont community composition might confer bleaching resilience to coral, so they could cope with different climate changes or potential future bleaching events. During this presentation, I will only displaying the first 2 top questions of this topic. And now I want to move on to what we've done last year in 2022. So in 2022, we decided to explore the coral reefs of a small island called Mayotte. It is located in the southern hemisphere, in the northern part of the Mozambique Channel near Madagascar. It is one of the least known regions of the world from the Indian Ocean. Mayotte Islands got a bleaching season that usually occurs during April every year, whether it be a massive bleaching or a minor one. And in 2022, it was characterized as a non-bleaching year. So we decided to focus on a specific coral species called Porites lutea, which is characterized as a massive coral species, so most of the time hemispherical, but is pretty resilient to coral bleaching. The aim of our project is then to explore what was happening before, during, and after potential bleaching event, or during the bleaching season. So I also want to introduce you to 2 key notions that I will speak during this presentation. First is the location on the reef notion. Our study will focus on a typical reef, which is called the fringing reef. And this kind of reef are divided in 3 compartments: the crest of the reef, which is near to the open sea; the inner reef, which is right in the middle, closer to the shore; and then you have the back of the reef, which is really right next to the beach itself. Usually in this kind of fringing reefs, the back and the inner parts of the reef are usually exposed to at low tide, so they're exposed to direct air and sunlight as well, while the crest of the reef is usually still underwater, even at low tide. The second notion that I want to explain is something which is quite simple, is the sun exposure. In the southern hemisphere, just like in the northern hemisphere, the sun rises from the east to the west. But at one point, there is a difference between northern and southern hemisphere, where at midday, the sun is pointing north in the southern hemisphere. Unlike in the northern hemisphere, it will point the south. And this affects the sun exposure for coral as well, where, for example, on that picture, you will see, you will observe that the sun exposure is much more important on the north part of the coral colony rather than on the south part. So this, when I refer to orientation later on, I will mean the orientation to the sun exposure. So on the island of Mayotte, we selected 5 sampling sites across the island, and on each site we picked up 15 coral colonies, which are located and divided in 3 groups of 5 in the back of the reef, the inner part of the reef, and the crest of the reef. So we have good representation of what is happening at the scale of the reef for each of our sites. In total, we selected and GPS tagged across the island 75 coral colonies. And on each site, we also inputted some temperature logger for each site to get the precise temperature for each of the sites across the island. And we also repeated the sampling one time in April, one time in May, and one time in June, in order to have a time series across the bleaching season in Mayotte. So what was the sampling procedure for our project? We wanted to collect coral tissues, and we used syringe sampling methods in order to harm as less as possible the coral and sample as best as we can the coral tissue in the north, top, and south of each colony on each side. So for one colony, we will do 3 different samples, one on the north, one on the north and south, to get the, like I said previously, the sunlight impact on the colony. Once this density once this sampling was done, we moved on to the density measurement, where for each coral tissue sample, we've got centrifuge, and we then placed 5 microliters from a sample on a grid microscope slide in order to count the density. And this grid looks exactly like this, and we selected 5 rectangles for each sample to make a good density estimation that we then inputted and you count manually using a specific software through ImageJ. And we then compile them into one raw dataset with different parameters, which was the site, the number of the colony, the location on the reef, and also the orientation, so the orientation against the sun. And then we then displayed them by months for April, May, and June. So using that dataset, we moved on to the proper work from these datasets, where the first result would display much more of the special pattern aspects. So in that slide, you can see a bar plot representing the mean symbiont density and standard deviation displayed by SAL variable. And as you can see with the letter above each bar, we do have a significant difference in terms of symbiont density present between sites, which means that there is a density data are driven by the geographic location of our samples. When we move on to a thinner spatial scale, when we move on to the location on the reef, so with samples that were close to the beach or close to the open sea, there seem to be no significant differences between the 3 locations within a reef. So the fringing reef does not influence, the location on the fringing reef does not influence our density data. And then when we move on to the northern, thinner, again, special pattern, which was the location on the colony to explore the potential effect of a sunlight exposure, we also saw no significant impact of the sun exposure on the density data. So there is no sunlight exposure on the gradient from north to the south of the colony, including the top. And the last aspect that we wanted to explore through that was the temporal aspect, as we sampled through April, May, and June. And on that topic, we saw a significant difference between the sampling month, which suggests a seasonal pattern on the density of symbiont within corals. The thing is, for the past 4 graphs that you've just seen, is they are only explore one data set on a univariate analysis. And as coral reefs are quite a complex structure, they need to be explored in a multivariate aspect. So we went on to a non-metrical multidimensional scaling. And this is a graphical representation of this new ordination of our dataset, which allow us to compile different variables together and see how they interact with each other. And as you can see here for the time effect, we can see that the community ordination seems to graphically follow an effect of time variable, just like in the univariate analysis. So there is a difference between April, May, and June sampling. And you do find the same pattern when we move on to the site aspect, where we have this kind of split between two groups with sites that are behaving differently and following different pattern, just like in the univariate analysis. But then when we move on to the location on the reef, we also explore that aspect and see that there is no graphical differences with the univariate approach, as well as for the orientation variable, where the sun exposure doesn't seem to have a direct impact on our density data in a multivariate analysis. But from that graphical aspect, we wanted to see if this trend that we see on the multivariate graphics were followed on the statistical analysis behind that. So we went on for a permanova to test the significance, if there was a significant difference between all variables. And it appears that we have a significant difference between three sites on our data set, and all of them are located on the west side of the island. And when we move on to the orientation variable, so the orientation to the sun exposure, we have known significant differences with density at the top of a colony with the south of a colony. So there is very suggests that there is a difference of significant density between the top of a colony and the south of a colony. And on another note, the location on the reef is still not significant between the different compartments of a fringing reef. So there is no difference between the back, the inner, or the crest of the reef. So in first conclusion to my question so far, from a spatial perspective and between sites, there is a significant, as we said, there is a significant difference between sites across the Mayotte island. And like I said, 3 of them are located on the west side of the island, while the other 2 are on the east side. And according to fishermen knowledge, local fishermen knowledge, there is different kind of reef fishes between the west and the east side of the island. And these, some reef fishes are known to be grazing the coral reefs. So this might attack the coral tissue itself and impact the density of our symbionts. Another explanation could be the topography of our sites, where some sites are much more open field and open to waves and wind. And on the other hand, we do have some sort of base sites that are much more shaded and not impacted by waves or high wind during the windy season. On the scale of a site, we've seen that there is like no density pattern between based on the position of the fringing reefs. So this suggests that the density that you can find on the back of the reef is the same as the one that you can find on the inner and the crest of the reef. So there is no significant differences between the two. So this is kind of homogeneous compartment at the scale of a site. And then when we move on to the individual aspect, much more within the coral, there is a sign of a density pattern for the sun exposure within the coral, as we observe much more density on the top of the colony rather than the south, which is linked to the pattern of the sun exposure in the southern hemisphere. So this might need more investigation to validate this observation, but we will try to explore that in the near future. And then there is also the temporal pattern, but based on our result, we clearly observed a temporal variation in the symbiont density And this happened even in a non-bleaching year, because like I said early in the presentation, 2022 was not a bleaching year. But in the other hand, you still have what we call a bleaching season, which occur in Mayotte, as like I said, in April. And we do observe a lower density concentration in April compared to May and June for the following months. So these are my principal answer to my question so far. And both next step would be the genetic analysis of our sample to make the link with the density result that we got. And as I said, coral reefs are quite complex models, so we want to add also much more temperature aspect using the loggers that we inputted inside, but also compare it with satellite sea surface temperature and also cloud cover that might affect the sunlight exposure. And we also aim to resample the colonies that we've done in 2022 next year, as 2024 could be a warmer year due to the potential return of the El Nino phenomenon. So I want to thank the University of Glasgow, MAST, and the University of Umeå for their financial and technical support. And also a huge thank you for the Centre Universitaire de Mayotte for their technical support on field and also in lab in their beautiful island of Mayotte. So thank you for your attention.
Thank you so much, Seb. A really interesting talk today. So if anyone has any questions for Seb, please can you put them into the Q&A box and we'll get round to them. So just while people are thinking of their questions, Seb, I was wondering if you could perhaps ask one of mine. And that is, why did you choose this coral in particular?
The aim of choosing this coral here in particular is that it is a massive coral reefs and it is pretty resistant to bleaching. We could have picked up a branching coral reef like you can see also on that picture on the left, but they are really sensible to bleaching and don't really have the capacity to recover from bleaching. As we want to study the effect of coral bleaching and how the coral behave after, we need a species that is pretty resistant to coral reefs and can recover, have the ability to recover.
Okay, thank you. So I think I might have time to ask another one. So, so what difference would you expect between sampling in a non-bleaching year and a mass bleaching year? I saw that you mentioned the El Nino.
Mm-hmm. Yeah. During a non-bleaching year, we observe some sort of seasonal variation of the density with like regular temperature. And what we might have during, for example, a year with a mass bleaching event is that the coral will expel most of its symbionts present within, and which will drastically reduce the density even more than in non-bleaching year. So we would probably have the same kind of pattern, but with a much more higher value, I would say.
Thank you. If anyone has a question, please just put it into the Q&A box. So Seb, I was wondering, because I'm not a coral expert, so I'm taking from this that bleaching is actually a natural thing that happens to corals, or is this something that's just started to happen due to human impacts?
Coral bleaching is a natural stuff that already happened in the past centuries. The only issue with that is that the coral bleaching since the 1980s, 90s is increasing more and more, and it's much more severe. So we have more and more bleaching events appearing throughout the globe. And the thing is with global warming in general, which is partially caused by human activities, that is warm the sea, warm the ocean. And this has a drastic impact on the coral reefs as they live really close to their temperature optimum. So if you have a changes of like 1 degree in the sea, it will affect directly the coral because they couldn't cope with these kind of changes.
Okay, thank you so much, Seb. If you just switch to the end slide so everyone can see your email. So if they do have any questions, they can just email you and follow up with you then.
Yep.
So everyone note down Seb's email at the bottom there. And just while I have you all, just so you to note that we have more talks coming up. They're every couple of weeks, and if you would like to attend any of those, then please sign up on the MASTS website. And thank you for everyone for joining, and thank you for Seb for a really, really interesting talk today. Thank you.
It was a pleasure.